move chlorine, which is negatively charged, to the negative end.
Is it hyperpolarized?
Hyperpolarized, right? Or what happens if you move a positive to the positive end? Hyperpolarized, right? So if you move a positive over here, this gets a little more positive, and this gets a little more negative, right? That's going to be hyperpolarizing. All of this assumes you are starting at resting membrane potential. We will move some of these things around after we have already depolarized, right? And that's going to change what happens a little bit, right? We're going to switch terminology. The phenomenon is the same, though, right? And you should be able to figure it out. So movement of potassium and chlorine in a resting cell is going to hyperpolarize the membrane. It's going to make it more difficult to generate an action potential, right? We're going to walk through the actual action potential today. We've kind of been dancing around this term. We're going to do this today, though. So the result of hyperpolarization is inhibitory. It's going to prevent the generation of an action potential in that cell, right? The book uses this big long term. I'm really more interested in the predictive ability, right? What happens to membrane potential as you move it over, right? So in hyperpolarization, are you going to get closer to zero or farther away? Farther away. So our example of an inhibitory synapse is going to be GABA, right? And I like GABA because this is one of the cartoons that my kids still watch every now and then.
Thank God I'm past that.
Yeah, my 4-year-old still does Yo Gabba Gabba every now and then. So it's this big long word, right? GABA is fine. So GABA is going to be moving around chlorine ions, right? So this is our neurotransmitter. This is our facilitated diffusion channel, right? We're going to call it a ligand-gated channel. It starts off as closed. The GABA binds to it. That opens the channel. Chlorine moves from high concentration to low concentration. Right? And then what happens to my membrane if I start off at resting membrane potential? Hyperpolarized. Right? You have to be able to walk yourself through these steps. So the specificity is really occurring here, right? GABA will only interact with this channel. This channel only allows chlorine to pass, right? And so that's where our specificity is coming from. So it's medically relevant. And again, as a microbiologist, I tend to go with microbiology kinds of things, right? So a good example of where this becomes significant is disease tetanus, right? Everybody heard? Y'all heard of tetanus before? I mean, I know that you have, and I know that you have, but what's kind of the common name for tetanus?
Lockjaw.
Lockjaw. Perfect. Right? What I was told as a little kid is, for whatever you do, don't step on a rusty nail, right? And it was always rusty, right? The rust has nothing to do with it. What it has to do with is the deep puncture wound that results. And if what is puncturing you is contaminated with soil bacterium, you run the risk of developing this disease tetanus, right? The toxin from the bacterium that causes tetanus blocks this exocytosis of GABA at these GABAergic terminals, right? Those GABAergic terminals normally inhibit muscle contraction, right? They actually keep your muscles from contracting. That's really important, as it turns out, because if you are contracting your biceps brachii, do you want to be contracting your triceps at the same time? No. Generally not, right? Because they'd be pulling in opposite directions, and that's not a good thing. So while this one is contracting, there's often some GABAergic terminal that's preventing this one from contracting, right? And that's really important. When you block this GABAergic terminal, right, what happens is you get rigid paralysis. Muscles contract and they cannot relax, right? They can't let go. And one of the early symptoms is lockjaw, right? It causes a rigid paralysis of this masseter, right? And it locks your jaw closed, right? eventually you can cause rigid paralysis all throughout the body, right? And you can actually get the agonist and the antagonist pulling so strongly at the same time, you can end up breaking bones, right? What kills you is this muscle here, though, right? What's this one here?
Diaphragm.
The diaphragm, right? Rigid paralysis of the diaphragm blocks respiration just the same as flaccid does. And again, it's all just back to this signaling event. So we don't die of tetanus in the United States very often. Why not?
Tennis shot.
Yeah, tetanus. You step on a rusty nail, you hump it down to the doctor. They say, well, I can't remember when the last time you did it, so we're just going to give you another shot. People do still die of tetanus. One of the biggest causes, one of the biggest kinds of tetanus that you see now, particularly with mortality, is when it happens to little babies, right? Because what happens is in countries without medical infrastructure, they clip that umbilicus with something that's dirty, and they infect the umbilical stump with the bacterium, right? And so this baby probably didn't live through this. Right? So now, so there's a lot of medical relevance to this, and we'll kind of expand on some of this stuff as we go along. But so one of the things that I have glossed over is that it's not just a single synapse. If you go upstairs and you look at those models, if you look at that model of the neuron, there are all kinds of different axons going into the dendrites in the body of that cell, right? And it's not just that one of them is signaling at a time, right? They are all signaling potentially at the same time, right? Some of those signals are going to be excitatory, right? Meaning they're going to depolarize. Some of those signals are going to be inhibitory. And so what the cell has to do is it has to make sense of that mess. What it's going to do is called summative, right? It's going to add it up. It's going to add up all of the depolarization, all of the hyperpolarization to get to one final membrane potential. And if that membrane potential depolarizes just enough, then you're going to generate an action potential, right? But this is very common. Most of your neurons are receiving information from multiple different stimuli at the same time. Particularly common in the central nervous system where you've got a whole bunch of interconnectedness of all of those neurons. So that brings us to this concept of a threshold potential. You don't have to depolarize all the way to zero in order to generate an action potential. You have to get closer to zero than your resting potential, but you don't have to get all the way to zero. Usually you don't even have to get that far, somewhere between minus 40 and minus 55, right? So if you're starting off at 60 or minus 70, right, that's not very far to get. So you're going to receive a whole bunch of signals at once, and if when you add it up, the region of interest gets to this threshold potential, that's going to start the cascade of events that generates an action potential, right? So we're going to have lots of inhibitory and excitatory signals at once. You're going to add it up, right? That's what summation means. It means addition, right? When you add it up, if you get to the threshold potential, then you will generate an action potential, right? So are you all with me so far? So where we're going to measure all of this is actually not where the synapse is. Where we are measuring the threshold potential is actually way over here at the base of the axon, something that usually gets called either the axonal hillock or the trigger zone, right? So we're going to be looking at the membrane potential not up here. We actually don't care about here. We're going to be looking at the membrane potential here. We're starting off at resting membrane potential, minus 60, minus 70, whatever. And then we're just going to say, can we get this all the way down to minus 55, minus 50, right? Whatever our threshold for that cell happens to be. So We're going to receive information at the dendrite of the cell body because that's where we said the axonal knobs are synapsing with the cell body or with the cell dendrites. We're then going to have an influx of ions. We're going to have those ions diffuse to the other end of the cell. Right? We're going to have the sodium that's leaking in through these ligand-gated channels diffuse this way. And if we can get enough over here of the right kinds of ions, meaning sodium or calcium, then at that point we're going to initiate the cascade. Right? So sodium diffuses that way. Right? Diffusion of ions, right? If it's the right kind of ions, will depolarize that adjacent membrane. It can only diffuse far enough to depolarize a couple of millimeters away, though, right? That's a major limitation. Because that means that I can get sodium over to about the base of the axon, but I can't actually diffuse sodium from here all the way to the end of the axon that would be down here somewhere, right? And these axons can be long. You have axons that are emerging from the central nervous system right around here and going all the way down to your big toe, right? And you have some that are coming all the way back up, right? Just a single axon from a single neuron. That is much too far to have this initial influx of sodium get all the way down there, right? And that's just us, right? Some of our axons are a meter long. Think about a giraffe who's got to run a single axon all the way from the base of its spine to the very tippy toes of its feet, right? Really long cells. So what we're going to do is we're going to have another series of channels that are gated channels that are going to open in response to a stimulus, right? The stimulus here is not going to be a ligand, though. The stimulus is going to be the voltage across the membrane, right? If you can get this voltage to minus 55, it's going to open this first channel. And then you're going to have stuff leak in. And that's going to depolarize enough to open this one. And then that's going to depolarize enough to open this one. Right? And it's going to be a chain reaction, one after the other. From the very beginning all the way to the end is going to open. Right? Are y'all following me on this? Does this make sense? All right. So, diffusion of ions, depolarize adjacent membranes up to a couple millimeters away. This is enough to get you to the base of the axon. Doesn't get you all the way down the length of the axon, though. So what we're going to do is we're going to depolarize this trigger zone within the axonal hillock. If you can get to the threshold potential, whatever that happens to be for that cell, that's going to open the floodgates. It's going to open this first little set of channels that are sensitive not to ligands but to electric potential. They are sensitive to voltage. So they are voltage-gated channels. This one opens, lets in sodium. This one opens, lets in more sodium. This one opens, lets in more. This one then will open, then, and so on and so forth, all the way down the line. So the voltage-gated channels are normally closed, right? That's a concept we should be okay with. They open when you reach threshold potential. They are present, generally speaking, in the axon, not in the cell body or the dendrites, right? This is the simplest way to architecture this. There are other ways to do it, right? I have a video. Your video gets confused as to how some of this stuff is working now. So be careful when you watch this video, right? So I'll point it out in a minute. So, resting membrane potential. Sodium on the outside, potassium on the inside. Your book in the video talk a little bit about how you set this up initially. Right? Your book will talk about something called the sodium-potassium pump. that will move sodium this way at the same time that it moves potassium this way. That's how you set up this initial distribution in a resting cell, right? I'm not going to ask you anything about that, right? But you may need to understand what's going on if you try to go back to the book. Resting membrane potential. Neurotransmitter docks opens the ligand-gated channels, lets in sodium. Sodium opens up the first group of voltage-gated channels. Right? Sodium moves in. You actually have a massive flux of sodium through these voltage-gated channels. Right? So much so that you can measure this spike. It goes all the way up to 0 and then some. Right? So once you open these voltage-gated channels, this is a major floodgate of movement of ions. Right? So you have a spike in depolarization. It only is going to last a very short period of time. Right? This is an important concept as well. Your neurons need to keep firing over and over and over again in order to generate a continuous effect, right? When you contract your biceps brachii, if you keep it contracted, your brain has to keep giving the go signal to your muscle. That means you have to send action potential after action potential after action potential. So after this depolarization occurs, you want to get back to threshold potential, or you want to get back to resting membrane potential almost as quickly as you can so that you can send a signal again if you need to, right? This ability to fire over and over and over again in rapid succession is very important to how neurons work. In order to repolarize, right? So when we depolarize, what did we allow in? Sodium, right? You could have used calcium, but we're using sodium for all of our stuff. When we repolarize, we're going to move potassium usually. We said that potassium is going to hyperpolarize if you start at resting membrane potential. We're not starting at resting membrane potential, though. We're starting depolarized. So what we're going to do is not hyperpolarize when we move potassium. We are going to repolarize. We are reestablishing resting membrane potential. Right? Does that make sense? The only thing that's different here is instead of starting at minus 70, we're starting at 0. We might even be starting at plus 1, plus 10, plus 20. Right? The effect of moving potassium from inside to outside, though, is going to repolarize the cell here. Right? So we have resting membrane potential. When we open the sodium voltage-gated channels, we shoot back up. This massively depolarizes the cells, and it can actually reverse polarity. That is going to open another set of voltage-gated channels, but these are voltage-gated potassium channels. And that's going to allow you to repolarize just as quickly. And sometimes you actually overshoot a little bit. This wave, right, that's your action potential. This wave is going to propagate all the way down the length of the axon. Right? It's a dynamic process. Very hard to get out of just looking at a still picture. That's why I'm going to do a video in a second here, even though the video has its own issues. The video is really important. What's up, mister?
You said so the potassium that goes out is also voltage-gated channels.
Yeah, those are voltage-gated channels. So the initial depolarization opens up the voltage-gated sodium channels. The further depolarization opens up the voltage-gated potassium channels so that it brings it back to where it was. Does that make sense?
Yes.
All right. This is very important though, right? This is, I know a lot of this seems like really overwhelming and unnecessary detail as to what's happening, right? This graph is very important though, right? Because what you were going to do in AMP2 and what a lot of you will end up doing in a medical setting is you will be looking at something that looks like that. What's that?
Heart rhythm.
That's a heart rhythm, right? That's a sinus rhythm. That's this thing right here, right? Except it's even more complicated because you've got multiple things that are depolarizing and repolarizing. But that's what you're looking at. You're looking at moving ions around within the cardiac tissue. Right? And what you will do as a clinician is say, well, this length of time is too long, right? You've got an elongated QT. Right? And that means that this problem is currently happening within that person's heart. But to understand what's going on here, you have to have at least a basic understanding of what the hell you are looking at, right? And it's going to get more complicated in the cardiac stuff that you do in AMP2, right? Because you're going to have different kinds of ions that are moving. You're going to be doing a lot of stuff with calcium instead of sodium. And you're going to be naming this. You're going to say that this is the P wave, and this is the QRS, and this is the T. Sometimes you'll even throw in a little bit of U there, right? And you're going to have to match this up to, well, one of these is atrial depolarization, one is atrial repolarization, one is ventricular depolarization, right? So on and so forth. Right? So you got to get this stuff down now. All right. So are there any questions before we push on? What's up, Malcolm?
All right, he said whenever it's repolarized, the potassium gets pushed out. Right?
Yeah. Movement of potassium from inside to outside will do one of two things. If you're at resting membrane potential, it will hyperpolarize you. If you are depolarized already, it will repolarize you.
All right, but I thought potassium was already present in the cell.
It is. So it's not moving into the cell, it's moving out of the cell.
Okay.
Okay, so that's what I've shown here. So we started off with potassium inside and sodium outside. So when I open the first set of voltage-gated sodium channels, that lets sodium in. That's then going to open up the voltage-gated potassium channels, which is going to move potassium out.
Does the potassium ever does it ever go back into the cell?
Yeah, well, eventually, yeah, absolutely. Eventually, you run out of sodium on the outside and you run out of potassium on the inside. And so eventually, you have to use this thing which I mentioned a little bit ago called the sodium-potassium, it's a sodium-potassium antiporter that's going to move sodium back outside at the same time that it moves potassium back inside.
Okay.
So eventually, you have to flip back the way you were. Absolutely. The video will mention that. But
I think he's talking about doesn't that little gate isn't it like three sodiums to potassium or something like that?
yeah. But that's not what we're dealing with, right? I'm actually not going to touch on that in the exam. I just want to get this initial wave through. Maybe I need to, in future semesters, deal with that a little more explicitly, but I'm already pressed for time this semester. All right, so this graph will absolutely be on the exam, right? Resting potential, depolarization, repolarization. Okay? Potassium is moving where? Here, here, here. In which A, B, or C, where is potassium moving from inside to out? C. Okay? Those are the kinds of questions you're going to need to be able to answer about this. So this wave of depolarization is going to move all down the axon, right? You're going to have an initial depolarization, and then you're going to have right behind it a repolarization. It's going to happen all the way down the axon, right? Okay, so that's what we're going to be dealing with here. The beginning of this movie gets a little confused about where the voltage-gated channels are and where the ligand-gated channels are. So I'm going to actually skip that initial part, and we're going to jump straight to the part that's accurate. But if you go back and look at this, keep in mind that usually, for the purposes of this lecture, ligand-gated channels are in the body and in the dendrites. Voltage-gated channels are in the axon. All right? We okay with that?
And the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential
That would be
and
the
to
answer to your
establish
question.
proper concentrations of sodium and potassium ions inside and outside the cell.
All right. So, let's do it one more time. All right. So, start off like this. Reach threshold potential. Allow sodium in. That decreases the polarity, right? It depolarizes the membrane. It depolarizes the membrane eventually so much that the voltage-gated sodium channels close, the voltage-gated potassium channels open, right? That allows potassium out. That repolarizes the membrane, right? All right, any questions? This is tricky. Have I hopelessly lost anybody at this point that wants to admit it? It requires sometimes going back and running through this process a couple of times on your own. So, wave of depolarization and repolarization all the way down the axon. Reaches the presynaptic terminus. That's going to release neurotransmitter into the synaptic cleft. And then what happens? Yeah, then you start the whole thing again in the next cell at the end of the line. Eventually, right, you will have an effect on the target, right? Target being gland or muscle. All right. At some point, you have to stop signaling, right? At some point, you don't want to contract that biceps brachii anymore, right? Or at some point, you want to turn off the secretion of that sweat. As long as this neurotransmitter is in the cleft, though, this neuron is going to keep on doing its thing as best as it can. So in order to shut off this signaling, you have to get rid of that neurotransmitter somehow. Two ways to do it. You can take this neurotransmitter and you can destroy it somehow. Or you can take that neurotransmitter and you can take it out of the cleft and put it usually back where it came from in the presynaptic terminus. You have to clear the cleft, though, in order to close these ligand-gated channels. Does that make sense? All right. Two ways to do it. The first way, you take it and you put it back into the presynaptic terminal. That's called reuptake. Right? And this is a term you will probably run into in medicine because there's a whole class of drugs that are reuptake inhibitors. And a reuptake inhibitor prevents reuptake of the neurotransmitter. And so it keeps those neurons on. Right? Does that make sense? The other way is destruction. Right? You somehow chemically destroy that neurotransmitter. Right? And this we can modify as well. So, lots of recreational drugs, right, which are bad for you, as it turns out, right, will affect your brain. They are psychoactive because they are affecting the amount of neurotransmitter in the cleft. Right? Cocaine is a classic reuptake inhibitor. You've got this neurotransmitter dopamine, right? So you've got all these dopaminergic terminals within your brain. Dopamine is associated with stimulating the pleasure centers of your central nervous system. They make you feel good. That's why people take cocaine. But again, when you do this, you are permanently sometimes altering the chemistry of your brain. Right? You are changing how your brain is working because what you're doing is you're turning on neurons that are not supposed to be on. Sometimes you can remove these effects, right? Although the result for a while is withdrawal because your brain has grown adapted to it. Sometimes some of the changes are irreversible. So, but it's all just modulating the signaling of it. Right? Prozac is another classic reuptake inhibitor. Have y'all heard of Prozac? I'm assuming most people have heard of cocaine. Have y'all heard of Prozac as well? Very common, right? There may be individuals who are using them. It is preventing the reuptake of serotonin. It is stranding serotonin within the cleft. And serotonin affects mood, right? And so this is a very classic antidepressant. There are lots of good reasons to alter brain chemistry. There are also lots of bad reasons to do it, though. And you have to be careful with anything that affects the nervous system. You have to say, is the good I'm getting better than the bad that might be done? MAOIs. Have you all heard of MAOIs before? Right? There is an enzyme called a monoamine oxidase that is present in your body. And monoamine oxidases destroy all kinds of different neurotransmitters. And MAOI is a monoamine oxidase inhibitor. It prevents the neurotransmitter from being destroyed. And so that means you have signaling where you wouldn't otherwise. This is another classic kind of antidepressant. Okay? Now what you will see in medicine and in pharmacology is that there are side effects for a lot of these neuroactive substances because it's not just a single terminal that is using this particular neurotransmitter. And so you are having effects on lots of different synapses throughout the central and peripheral nervous system. So there is a lot of pharmacology that's just dealing with, well, what terminals are being affected, and what does that mean? Okay, so now myelination. What kind of cell is that? Schwann cell. We'll deal with the other kind of myelinating cell in a little bit. So I've got an unmyelinated axon, and I've got a myelinated axon. Now I've got gaps between the Schwann cells and a myelinated axon. What's the gap called? That is a node of Ranvier. Now, this is a question on last semester's test that really threw some people, and I had a couple of students get really mad at me about this question. Okay, so I'm going to make sure that I clear the air now. Do you find nodes of Ranvier in myelinated or unmyelinated axons? Only in myelinated, because it's not just the naked axon, it's actually the space between insulation. So if I were to say node of Ranvier, myelinated or unmyelinated, node of Ranvier, nodes of Ranvier are features only of myelinated axons. So, faster or slower? This is faster. So we'll get into why today. They are much faster, right? Upwards of 100 meters per second for those of you who are metric system challenged. 200 miles per hour. We can move signals from here down to here. That's very important. You need a lot of near instantaneous communication in a lot of your neural circuitry. Not all, but a lot. There are unmyelinated axons which move significantly slower. But this is one of the big things that you can do to speed up signal propagation. Why? So we've got an unmyelinated axon. What kind of channel do we find in the axon of the cell? Voltage gated, right? We got it all the way down the length of the axon. And what I said was that we're going to open up these channels, but we've got to do it sequentially. We've got to open up this one first and wait for a little bit of sodium to come in order to open this one. And then we've got to wait for this one to let a little bit of sodium in order to open this. Right? And you've got to do it all the way down the line, one after another, after another, after another, until you get to the end. Right? It takes time. Not much time, but it takes a little bit of time to open this one up. And then a little bit more. If your axon's a meter long, right, that adds up. So this works, but it's not terribly efficient, and it's not all that fast. But eventually the action potential gets all the way down the end, right? It's like, and I stole this from Parsons, this metaphor, it's like running down this hallway out here and stopping to open every single door along the way. It doesn't take long to open a door, but if you add it up all the way down the hall, then it adds up. In a myelinated axon, we only have our voltage-gated channels within the nodes of Ranvier. So there are fewer doors to open up along the way. And so our action potential is not going to propagate continuously all the way down. It's going to jump from node to node to node to node. And that, as it turns out, is much, much faster. Right? Because you don't have as many doors to open. Because you've got fewer channels. If you only open every other door, it's much faster getting from one end of the hall to the other. Right? Does that make sense, at least on a metaphorical level? There are other things at play here, but nobody's got a good grasp of physics or chemistry for the most part. But I can expand on it if anybody wants me to after class. So this is called saltatory conduction. There are very few benefits from children, but one of them is that I was exposed to Dora the Explorer. And what Dora taught me was that salta means to jump, right? And so saltatory conduction is this jumping of the action potential from node to node to node to node, right? So saltatory conduction, does it happen in a myelinated or an unmyelinated axon?
Myelinated.
Myelinated. It's got to be, because it requires the presence of those nodes of Ranvier. All right. Very important. Multiple sclerosis demyelinates a lot of the axons within the central nervous system. Slows down communication. It's damaging the myelinating cell within the central nervous system, this big long thing called an oligodendrocyte. What that means is that because everything is wired into your central nervous system, if you slow it down, you have all kinds of issues that result from this. And multiple sclerosis is a very, very debilitating disease as a result. Right? There are lots of other things that can cause myelination issues, including a lot of infectious disease potentially. So, all right. I really, really mean it this time. We are all done with the chemistry. Right? So, I know I've told you that 2 or 3 times now, but I really, really mean it. All right. So, now on to the easy stuff. Neuroglia. Right? We're going to see different kinds of neuroglia depending on where we are. Right? Where we're going to start with is the central nervous system. And the central nervous system is much more complex. And that means lots more neuroglia. This one here, right? This is the heart of the nervous system. This is what's doing all the signaling. The neuroglia has ancillary roles. Right? One of them we've already talked about. One of them is myelination, right? Insulation of the axons. Lots of other things that need to be done. The neuroglia are just accessory cells. They do all of the stuff that the neurons can't do for themselves. Neurons are great big babies and they cannot take care of themselves. So they need all of these other accessory cells. There are lots of other accessory cells. We're not going to worry about a lot of them. Right? We're going to constrain our list of test questions. Astrocytes, right? This is central nervous system only. Maintain the environment around the neuron. That's kind of vague, right? Astrocytes do a lot of the basic functions for a neuron. Make sure their environment is clean and sanitary. Your book doesn't really go into what this means, and so we're not going to worry about it. One thing that is important is this concept of a blood-brain barrier, though, right? Neurons are very fragile, and they do not repair damage very well at all. Have you all learned at some point that neural damage is forever, basically?
Yeah.
That's not entirely true, but it's not that far off either. Neurons just do not heal very well. So, as it turns out, we ingest or we breathe in or we are exposed to all kinds of noxious crap. And that stuff goes straight into our bloodstream and is transported through all of our body, right? And if you pick up some stray chemical that kills an epithelial cell, who cares, right? You just make another epithelial cell. You cannot do that with neurons, though. You have to make sure that they are there, right? So you've got a whole filtration system that's designed to filter out all the noxious crap from our blood and keep it from getting into our neural tissue. Right? One part of this is your astrocytes. They've got these feet that wrap around a lot of the blood vessels that are in the proximity of neural tissue within the central nervous system. And they make sure that the only stuff that leaks out of those blood vessels is stuff that you really want there. Glucose, right? Water, those sorts of things. Right? You need lots of ions from the blood, obviously, because you're moving stuff around. So, that's this concept of a blood-brain barrier. It's called brain, but it applies equally well to the spinal cord as well. The other half of the blood-brain barrier is these ependymal cells, right? They look like epithelia. They're polar. They've got these little things sticking off them. What are those little things called if you're dealing with epithelia? Yeah, not pili would be microorganisms. Right? This is cilia. Right? So, but they're actually, despite the fact that they've got these little cuboidal shapes, right? They really do look like epithelia, but they're not actually epithelial cells. These are nervous tissue, right? So this is neuroglia. They line a very special set of blood vessels called the choroid plexus. We'll see that in another slide or two. And they filter the fluid from those very special capillaries, and they use it to produce this resulting cerebrospinal fluid. Right? Where have we talked about cerebrospinal fluid in the lab? Where do you find it?
Oh,
Yeah.
no,
Well, no, that's correct though.
But the
Yeah, we haven't gotten there. Well, you've gotten there because you're a lab ahead of the rest of these people. But yeah, so you see it in the central canal of the spine. Right? You also see it between the arachnoid and the pia mater in that subarachnoid space. Right? This is being made by the filtration action of these ependymal cells filtering out these particular blood vessels called the choroid plexus. We will talk about them in a moment in their particular location within the ventricles of the brain, which I heard somebody read to me just a second ago. So, our myelinating cell of the central nervous system is not a Schwann cell, right? It's an oligodendrocyte. If you see a Schwann cell, it cannot be a peripheral neuron, right? So that means what's our model upstairs? Central nervous system or peripheral nervous system? It's got to be peripheral because it's got a Schwann cell. You've got a lot more neurons within a given amount of space in the central nervous system. That means you need to be efficient in your use of cells. And so what you see is that an oligodendrocyte has a body that is actually completely removed from the myelination, right? That's different from Schwann cells. And they've got these little feet that go around and wrap around the axon, right? And form the myelination. And they're not just myelinating a single axon, right? They're myelinating several axons at a time, right? So, and it's just because you've got more neurons in the central nervous system, and so you have to deal with things economically, right? You have to be frugal with your resources. Okay, so that's all the cell biology we're going to do. Your book talks about other things like microglial cells and other things. We don't care about those. So we'll come back when we get to the peripheral nervous system on whenever we get to it. Probably, what's today? Is today Tuesday? Probably not until Tuesday of next week. We'll come back and talk about Schwann cells and then something called a satellite cell. So, central nervous system. We're going to divide up into bits, right? You got the whole of the brain. You've got a subregion of the brain here called the brain stem. We'll talk about another subregion today actually called the diencephalon. We've got this transition of the brain stem into the spine. How do you tell the difference between the brain stem and the spine?
spine.
Yeah. No, that's close. It's location, basically. That's the only real definitive marker between the medulla oblongata and the spine.
Oh, yeah.
What's this hole that it's passing through? Yeah, the foramen magnum. If it's above the foramen magnum, it's brain. If it's below the foramen magnum, it's the spine. We're going to go into detail about some of these structures and some of these regions on Thursday, right? We're going to do a little cursory stuff today, and really we're going to talk about the ventricular system today. We've already done meninges in the lab, right? So I'm assuming everybody's okay with this material at this point. When do we get out of here? 2:15. Dura mater, arachnoid mater, pia mater. Most superficial, most deep. We okay with this stuff? Alright. We said that this is our major protective layer. You're actually going to be cutting this stuff off with scissors potentially when we do the dissections on Thursday. It's actually continuous with the periosteum in some places of the skull. It doesn't actually merge with the periosteum though in the spinal column until actually the periosteum has become wrapped around the nerves and is continuous with the epineurium, right? It's going to actually separate functional regions of the brain, right? We'll talk about in the lab today and in a lecture on Thursday about how you've got two hemispheres of your cerebrum, right? Well, the dura mater is actually going to fold in between them. That's going to be a problem during that dissection if the meninges are still there because that dura mater is really wedged in there and you're going to have to get it out somehow. You're also going to have a fold of dura mater that separates the cerebellum from the cerebrum as well, right? So we'll deal with all of that soon enough. So in the picture of your book, the dura mater is in the white. It's the most superficial. Wraps around the brain, folds in, separates some of the regions. Cerebellum here, cerebrum up here. This is all brain stem, and then transitioning into the diencephalon. Right? Goes all the way down around the spinal cord as well. The next layer down is the arachnoid mater. This means spider because there's going to be a web-like extension into some of that subarachnoid space. The space between the arachnoid mater and the pia mater is the subarachnoid space. This is where we've got a whole chunk of cerebrospinal fluid. Arachnoid mater in purple on this diagram. Right? The pia mater is the deepest layer. It is heavily vascularized. You will actually be able to see some of these blood vessels when we do the dissection in the lab. It is stitched on to the surface of the neural tissue. When you do the dissection, you will peel off the dura mater and the arachnoid mater, but you will not be able to separate the pia mater from the rest of the nervous tissue. It is literally stitched onto that stuff. Okay? Intimately associated. It is actually going to follow a lot of the wrinkles of the cerebrum inwards. Right? It's around the spine as well, right? And we saw the spinal cord section, and it should have looked like this. What's this region here that's above the dura? That's the epidural space. All right. So, the ventricles of the brain. What you're going to do is you are going to, when you do that dissection, you're going to take a scalpel or a knife, and you are going to do a mid-sagittal cut. Right down the middle. And you are going to, in the process, cut through these big cavities that are called ventricles. And then you'll be able to peer into some of the larger ones that are actually still preserved on the inside of either half. Right? These spaces within the brain are called ventricles. They're not empty. There's cerebrospinal fluid within them, right? And this is actually the site of manufacture of most of this cerebrospinal fluid, right? 4 ventricles. We've got the first and second ventricles, which are called the lateral ventricles. We've got the third ventricle and the fourth ventricle. These can be a little, right? These are easy. These two are subtle when you're doing the dissection and when you were looking at some of these models, right? So this is the brain, cerebellum back here, so this is posterior, right? This is anterior. You've got these great big horns, right? These are your lateral ventricles. They connected to this region here, this is the third ventricle, via this little tube called the interventricular foramen. The third ventricle drains down into the fourth ventricle here through the cerebral aqueduct. You might actually see the aqueduct if you're careful in your dissections. And then ultimately the fourth ventricle drains down into the central canal of the spine. Right? So, a couple different places you're going to see cerebrospinal fluid. Ventricles of the brain, central canal of the spine, and then the subarachnoid space. This is from a profile. Lateral ventricle here. Third ventricle here. Right? And then draining down into the fourth ventricle down here. Okay? So, cerebrospinal fluid is made within these ventricles. It's blood-based. The blood vessels are filtered by these ependymal cells that line the exterior blood vessels. Right? Present within the ventricles, right, where they're made, and then ultimately drain into the central canal of the spine and ultimately back into the subarachnoid space. The function of the cerebrospinal fluid, anytime you got fluid, right, I told you basically think two things. Lubrication, shock absorption. Often it's both things, right? The major one for cerebrospinal fluid is shock absorption. Since it's blood-based, it's also going to be supplying nutrients to the brain and transporting waste out of the brain, right? And out of the rest of the central nervous system as well. This shock absorption stuff, right, has gotten some news lately, right? There's been a lot of talk in the last year or so about brain damage that is the consequence of repeated injury to the head. You see it very clearly in boxing, right? Usually you can tell somebody who's had a long career in boxing because they start to have cognitive decline. Right? The big scary thing recently has been that there's been evidence that this is cropping up in people who play football as well. And the younger you start playing, the more risk there appears to be. Right? Normally when you shake your head, right, this cushion of fluid around the brain keeps the brain from slamming into the walls of the cranium. Okay? But in a much more impactful event like you would have in football, the brain ends up slamming into the walls of the cranium and doing physical trauma to it. And I think the term that they use for this now is chronic traumatic encephalopathy. Right? And what it means is just an accumulation of damage due to trauma, physical trauma to the brain. And so there's some concern that we don't have adequate safeguards for all of this stuff yet. So, made within the ventricles, you've got, we'll name all of these structures today in the lab and we'll come back to this stuff in the lecture on Thursday, right? You've got a little membrane here. What you will do in the dissection is you will slit this membrane and you will actually open it up and peer back into the lateral ventricle, which would be on the other side. When you do your cut, you're going to slice clean through the third ventricle. So the third ventricle is this space right here. Drains down into the fourth ventricle, which is the space in front of the cerebellum, right? Ultimately down into the central canal of the spine. So, lining these ventricles, you've got these set of blood vessels called the choroid plexuses that are lined with ependymal cells, right? This is where the actual filtration into cerebrospinal fluid is occurring. Regions lined with ependymal cells make most of the central cerebrospinal fluid present in all four of those ventricles. This stuff circulates. You have a flow from the ventricles into the spinal cord. It also will drain into the subarachnoid space, come all the way back up. Ultimately, it's going to drain into the venous system, right, back into the circulatory system. This ensures that you get nutrients to the brain and that you get waste products back into the circulatory system to ultimately be expunged through the urinary system. CSF from the ventricles flows in the central canal into the subarachnoid space. This is true of both the spine and the brain. It's going to flow out. So what you'll see is that in certain regions you have the purple one, which is the arachnoid mater, penetrate through the dura mater into what is this large vein. So this penetration is this wrinkly mess here called the arachnoid granulations. Very permeable membranes here, so they just allow movement of fluids and filtration from one side to the other. They flow into this large venous return called the dural sinus. It's called the dural sinus. What sinus? Cavity. The dura mater actually splits here, and the space between this split dura is the dural sinus. This split is only seen in one place. You can follow it right down the middle, right? So you only have it in the mid-sagittal line, not off to either parasagittal side. Right? This dural sinus runs from the middle all the way back. This is a coronal section. Right? That's going to be a problem with these brains. You have to be able to visualize these things from different cuts. This is a coronal section. This is the actual brain. This is the pia mater, the arachnoid mater, subarachnoid space. This is a fold of the dura mater in between the two hemispheres of the cerebrum. Above them is where you have this large dural sinus, where you have the arachnoid granulation piercing through the dura mater into the actual sinus. And this is where you're going to dump waste back into the veins so that they can be then expunged through the kidneys. All right. So it's kind of dry. Very important functioning of the brain. Dural sinus, big vein, essentially is returning stuff to the bloodstream. Okay, your book does a pretty good example of the circulation from the ventricles down into the spine, back into the subarachnoid space, back all the way up into the dural sinus. Right? It actually goes all the way down into the spine, though, right? And then it comes all the way back up. This is being passively circulated. You are constantly making new cerebrospinal fluid and circulating it all around your central nervous system. So one of the interesting things here is that the spinal cord stops before you get to the end of the vertebral column, right? And a lot of people didn't realize that coming into this class. The spinal cord stops usually in the vicinity of the first or second lumbar vertebrae. And then you just have this cluster of nerves coming down the rest of the way and ultimately innervating things down here. Okay? That's why if they want to sample your cerebrospinal fluid, they'll do it way down here where you're less likely to accidentally drive that needle into your actual spine, right? Because that's probably not a good thing. Anybody ever do a lumbar puncture or spinal tap before? No. I've heard it's extraordinarily painful. What do they do it for?
infection.
Yeah, well, sometimes they'll do it because they're wanting to drain fluid out. They also use it diagnostically. What do they use a diagnostic for? Yeah, meningitis is a big one. Ideally, you want it to be this kind of clear fluid, right? If it's yellow or cloudy, that's usually a bad sign. So, but again, we stop a little bit early, and so you can jab a needle into this space here. What's this space here? Yeah, the subarachnoid space. So again, there's a reason why you have to deal with all this junk. All right, that seems like quite enough for today.